Research from the American Heart Association. He's been elected to the National Academy of Sciences, the American Society of Clinical Investigation, and the Association of American Physicians. And he serves on many editorial boards and is the editor-in-chief of Molecular and Cellular Biology. The last bit of groundwork is that if you have any questions that pop up along the presentation, you can type them in the chat, and then we will address them at the end during a brief Q&A session. So with that, I'll hand it over to Dr. Tontinos for his presentation on new pathways in cellular and systemic lipid homeostasis.
Thank you very much. Can everybody see the slides and hear me? So far, so good? Okay. So by way of introduction, my laboratory is interested in lipid metabolism, and we try to elucidate mechanistic pathways whereby cells and animals control appropriately how much lipid they have and what different types of lipids they have, and to prevent both to maintain lipid homeostasis, but also to prevent lipid toxicity. So here are a couple of different mice. One of them is not like the other, and you can clearly see that there's something wrong here with the mouse on the left. And what this mouse has is massive accumulation of cholesterol under its skin. This mouse has about 5 times more whole body cholesterol than the mouse on the right. And that's a consequence of inactivating mutations in the LXR alpha transcription factor and the ApoE gene. So this is a mouse phenocopy of a characteristic lesion that's found in human disease, the disease of familial hypercholesterolemia. This was the disease that led to the discovery of the LDL receptor and the working out of the cholesterol pathway by Nobel laureates Mike Brown and Joe Goldstein. But the point is that there are pathways that operate within cells and within the body to maintain appropriate levels of lipids. And when these pathways go awry, they lead to pathology both in mice and in people. So 20 years ago or so now, my laboratory set out to answer a relatively simple question. So based on that mouse that mouse model that we generated, the obvious conclusion is that the LXR transcription factors, hopefully people can see my arrow here, up at the top, these transcription factors are clearly important for the control of lipid homeostasis, because if you delete them, you get a mouse that looks like that. The next obvious question is, well, why? Presumably, this transcription factor regulates the expression of a series of genes that help the animal maintain lipid homeostasis. And the simple question that we've asked over the last 20 years is, what are the genes that LXR regulates that are important for controlling lipid levels in cells and animals? And over the years, we've identified a number of different factors that contribute to the regulation of cholesterol, fatty acid, and phospholipid homeostasis in cells and animals, and have characterized a number of different mechanistic pathways. Today, I'm going to focus on our most recent discovery, and that is of a new family of cholesterol transport proteins called the asters. So by way of introduction to this one specific story, I want to remind you, maybe you know this, maybe you don't, but different membranes in the cell don't contain the same amount of cholesterol. In fact, cholesterol is highly compartmentalized within cells. Most of the cholesterol lives in the plasma membrane. Plasma membrane is about 30% by mass cholesterol. Whereas the endoplasmic reticulum, the ER, has among the least cholesterol of any membrane in the cell, only of about 5% cholesterol. Now that immediately presents somewhat of a conundrum for the cell, because all of the machinery that the cell has to respond to changes in cholesterol live in the ER. If a cell has too much cholesterol, it protects itself in two different important ways. Number one, it shuts off endogenous cholesterol synthesis by turning off S or BP. It turns on the LXR genes, which help the cell pump the cholesterol out. And finally, if this cholesterol continues to accumulate, what the cell does with that free cholesterol is to turn it into cholesterol ester through an enzyme called ACAT, which also lives in the ER. And storing cholesterol in lipid droplets is a much more safer way for the cell to store the cholesterol than as free cholesterol, which can be toxic. But if most of the cholesterol is in the plasma membrane, and the ER is where the machinery to respond to changes in cholesterol lives, that implies that the ER has to know what's going on in the rest of the cell at all times. And you can clearly show experimentally that this must be the case, because if you simply artificially put cholesterol in a cell membrane in a dish, you can do this using a vehicle called cyclodextrin, where you just add this to the cell. That puts the cholesterol in the plasma membrane. Immediately you can see within minutes the ER start to respond to that challenge. So there must be communication between these two compartments. But it's been unclear what that communication is. Now, to follow the talk today, you need to recognize one other subtlety about the cholesterol in the plasma membrane, and that is not all the cholesterol in the plasma membrane is the same. There's actually several distinct pools. The pool that we care about today is this so-called accessible pool, and this is a concept that was really worked out by Arun Radhakrishnan and his colleagues at UT Southwestern. And what they very nicely showed is that it's only this small accessible pool that's really in equilibrium with the ER. And that nicely explains why cholesterol is not constantly flowing downhill from the plasma membrane to the ER. If there's 30% here and 5% here, why isn't cholesterol constantly moving to the ER? The answer is that most of this cholesterol in the plasma membrane is not accessible. It's actually not in the equation. It's bound up with either other lipids or proteins, so it really doesn't factor in. It's sequestered. What we really care about and what the equilibrium really is here is between this small accessible pool and the ER. And if you raise this accessible pool, then the cholesterol will move to the ER. But if this accessible pool is below 5%, even though you have all this other cholesterol here, there's no transport. Now, there are two theoretical ways that cholesterol can move between two different membranes. One is through the so-called vesicular transport pathway. And so this is an example of this is the LDL receptor internalization through clathrin-coated pits. These vesicles go to lysosome, eventually the cholesterol is liberated. So one way to move lipids around cells is to have vesicles butt off one membrane and fuse with another. But it was proven decades ago that you can poison all vesicular transport in the cell and the cholesterol will still move very rapidly from the plasma membrane to the ER. So vesicular transport's not required for this pathway. That implies that there has to be a so-called non-vesicular lipid transport mechanism. And the simplest way to think about this would be a protein which helps move lipids from one membrane to another. The good news is that if you look in the genome of mammalian cells, there are lots of proteins that conceivably could be lipid transport proteins, and you would speculate that one or more of these should be involved in the movement of cholesterol from the plasma membrane to the ER. These include 15 so-called start domain proteins, 12 so-called OSBP proteins. But the difficulty in this field of cell biology has been it's been challenging to assign biological functions to these many putative lipid transport proteins. And that's because by and large, when people have done loss of function studies, at least so far, the mouse has not given a phenotype which has really clearly told you what the protein does. And there are two explanations for that. One would be that there's a lot of redundancy here, and maybe there's 4 or 5 proteins that all do the same thing, so that when you knock one out in a mouse, you don't get a phenotype. An alternative explanation would be that no one's done the right experiment in a mouse to uncover the phenotype, and then the third possibility would be that maybe these aren't, in fact, the right proteins in the first place. The one lipid transport protein had previously really has a very compelling phenotype in mouse is the original start domain containing protein, which is called STAR-D1. And this gives a very striking phenotype in both mice and humans. And the phenotype really gives you a strong clue as to what the biological function of this protein is. So STAR-D1 deficiency causes adrenal cholesterol accumulation, and it causes a disease called adrenal lipodystrophy. The adrenal gland is the small gland that sits on the kidney, and what it does is make steroid hormones, including cortisol, in the adrenal cortex. The final steps of that synthesis pathway happen in the mitochondria. So in order to make cortisol, which is derived from cholesterol, you need to import the cholesterol into the cell. You store it as lipid droplets, and then the lipid droplets are hydrolyzed. The free cholesterol enters the mitochondria where the steroids are made. The phenotype of the knockout mouse is that you get massive accumulation of lipid droplets in the cell, which implies that this cholesterol is not getting into the mitochondria. And in fact, what the function of this STAR-D1 protein turns out to be, it's a transporter that imports cholesterol into the mitochondria. So that phenotype clearly tells you what the protein does. And start domain proteins contain a very characteristic lipid binding fold. If you're a structural biologist, hopefully you appreciate this. If you're not a structural biologist, all you need to appreciate is that there's a cavity in here which is designed to accommodate a small molecule hydrophobic lipid. And there's a number of these proteins that transport different types of lipids. We became interested in this area of biology with the discovery of these three proteins, which we've subsequently called astroproteins because that has some relation to the function that I'm going to tell you that they perform. But at the time we became interested in them, they were annotated in the genome as three basically unknown genes, gram D1, A, B, and C. There were no papers on any of them, and if you simply blast the sequence against the genome, what you get are the two other proteins. So there wasn't much clue from the primary DNA sequence what the function of these proteins might be. They had a transmembrane domain on one end, a domain called a gram domain on the other end, which stands for 3 or 4 proteins that have a domain that looks like this, but by and large, they didn't really look like anything. And the key insight that really propelled this project forward was made by my MD PhD student, Jaspreet Sandhu. And the teaching point here is that all of us as PIs really depend upon the people who work for us. We try to inspire them, but at the end of the day, their own insight and their own drive is really critical. And it was Jaspreet's creativity that really launched this project. I'm not really a computer geek, and so I certainly don't sit in front of my computer, blasting or comparing sequence homology. That's just not something that I spend my time doing. I depend on the people in my laboratory to do that type of thing. And so Jaspreet took it upon himself to try to figure out if he could find something that these proteins looked like. And what he decided to do was to plug the DNA sequence into a structural biology secondary structure prediction program. And if you do that, what the computer tells you is that the middle of this protein actually looks like a start domain. So the lipid binding domain from the STAR-D1 protein. It looks like it in the way it folds, but the primary sequence is not at all. You would never be able to line it up. And that immediately implied that these were likely to be novel lipid transport proteins, which nobody had characterized. The LXR transcription factors are regulated by cholesterol. They bind oxysterols, which are modified derivatives of cholesterol. And Occam's razor in my laboratory is that any proteins whose expression is regulated by LXR must have something to do with cholesterol metabolism. So since we knew that the AstroB protein is regulated by LXR, the assumption from the initial stages of this project were that this protein must be a cholesterol binding protein and likely a cholesterol transporter. Usually when you make such broad assumptions early on in the project, they turn out to be wrong, and that's the way science works. But in this case, that simple assumption at the beginning turned out to be correct. But I'm going to walk you through the series of studies that Jaspreet did to prove his hypotheses. To prove that this was a lipid binding protein, what he first did was purify that central domain by itself, and then develop a binding assay where he could report the binding of small molecules to that domain. We used a fluorescently tagged cholesterol molecule, and he showed that if you put the tag on this end, it very nicely binds the 3 aster lipid binding domains, but if you put the tag on this end, you get no binding at all. So that's very nice. It tells you there's some specificity to what can be accommodated in the pocket. And these domains will also bind native cholesterol itself, which is likely to be the endogenous ligand. All right, so those series of studies proved that the proteins bound cholesterol, but just because you're a cholesterol binding protein doesn't mean you're a cholesterol transport protein. The LXRs bind cholesterol derivatives, but they're not transporters, they're transcription factors. So how do you establish that your protein of interest actually is a transporter, can move cholesterol from one membrane to another? The first way we did that was in a test tube. We made 2 artificial membranes, one small and one big, and you can make them different densities so that at the end of the experiment you can separate them by centrifugation. And then what you do is you put cholesterol in one and not the other, and then put them together in a test tube and throw in your protein of interest. And if you do that without any transporter, you can leave the 2 vesicles on your benchtop for the next week and a half, and the cholesterol will never move because it cannot just float through aqueous solution, and even the membranes bumping into each other doesn't really move cholesterol at an appreciable rate. If you throw in any old protein into the test tube, such as albumin, nothing happens. But if you throw in any of the 3 essential domains from the aster proteins, it very efficiently catalyzes the movement of the cholesterol from one membrane to another. And that really proves that that central domain, even all by itself, is capable of transporting cholesterol between membranes. Presumably, it's extracting the cholesterol from membrane A, bumping into membrane B, and then depositing it there. We went on to solve the crystal structure of the aster central domain. And by we, I mean the team that we assembled to do this. And the other teaching point that I like to make here. So John Schwabe is a structural biologist at the University of Leicester in the UK. And one of the important things you need to do when you're running your own laboratory is figure out when you need to do things yourself and when you need to collaborate, because it's easier and more efficient to collaborate. I like to collaborate. I like to assemble teams. A challenge can be, well, who do you collaborate with? How do you find a structural biologist to engage in a fruitful collaboration? And the teaching point here is make friends with your fellow postdocs and grad students. Because the way we found John Schwabe to collaborate with was he was a postdoctoral fellow with me when I was in Ron Evans' lab at the Salk Institute. And we actually didn't work together. We drank wine together and went on small plane trips. He was a pilot, so we became friends. And since we were friends, I trusted him to collaborate with, and over the past 20 years or so, we've now crystallized multiple proteins together, him from the structural side and me from the biology side. So identifying good collaborators is important, and keeping your friends and making them along your road in science is important for success. So John was able to solve the structure of the central domain of the aster proteins. This here is a 25-hydroxycholesterol ligand embedded in the pocket. This is a glycerol molecule, which is also floating around here. This was probably, glycerol was present in the crystallography buffer, so this probably doesn't bind glycerol physiologically, but it does show that there's some extra room here in the pocket, which could be useful pharmacologically later on. Crystallographers like to make movies, so I'll show you a little movie. This thing will spin around, and what I want you to ask yourself while the thing is spinning around is, how does the ligand get in and out of the pocket? If you notice, the protein is really completely encasing the ligand. There's not an obvious in or out route, but we've now crystallized all 3 of the central domains of the astroproteins, and they pretty much all look like each other with one exception. And the exception is that this little red loop is actually in slightly different positions in each of the 3 proteins, which implies that it's floppy. So we think this is actually the door. So you can imagine that this door can flop open, allowing entrance to the lipid binding cavity, and then it can close again. And that way the cholesterol can be shielded from aqueous solutions at all times when it's moving around the cytoplasm. So that implies a mechanism for how the proteins work. We think this end of the molecule, which has a lot of nonpolar and also some basic charges here, which would be nicely situated to make contact with a positively charged phospholipid bilayer. We think this end of the molecule sort of bumps up or slightly inserts itself into the bilayer. The door then opens, allowing the ligand to come in and out, and then the protein can leave that one membrane and move to another membrane and deposit the cholesterol there. And that way the cholesterol can move without ever being in the cytoplasm. But clearly, if you were designing the system and you want to specifically move cholesterol from one place to another in a controlled fashion, you need more than just this central domain. Because as I showed you in a test tube, that central domain all by itself will move cholesterol. So if you had soluble central aster domain just floating around the cell, it would just move cholesterol randomly from one place to another and dissipate all the appropriate gradients. So you don't want that to happen. So you need a way to specifically move cholesterol at certain times and from one place to another. So that's what the rest of the aster proteins do. They have a transmembrane domain on one end, which presumably defines the membrane that can receive the cholesterol, and they have this other domain on the other end, which presumably defines the membrane that can donate the cholesterol. And so the rest of the other 2 domains really tell the aster molecule where to move cholesterol to and from and when to move it. So we set out to try to understand how this works. The first question we asked was, well, where are these proteins anchored? Where does this transmembrane domain live? This was the relatively easy question. We did colocalization studies, and it very quickly became apparent that these are all integral ER membrane proteins, all 3 of them. We cannot localize them to anywhere else except the ER. So then we can update our model. The aster proteins live in the ER, and presumably they're positioned to accept cholesterol from some other membrane. And we hypothesize that it's this Gram domain here, which is making contact with some other membrane. And then the question became, what is that other membrane? To address this, Jaspreet purified the N-terminal Gram domain and did isolated lipid binding assays to try to find a lipid it would interact with. And what he found is that this domain likes to bind phosphatidylserine, which is abundant on the inner leaflet of the plasma membrane. And that was an exciting discovery for us and immediately suggested that these could be, in fact, plasma membrane to ER cholesterol transporters, which people have been looking for quite some time. We were excited by that idea, and I challenged Jaspreet to, okay, go prove that these things interact with the plasma membrane. And you have to be careful as a PI when you strongly let the people who work for you know the answer that you want, right? So it's one thing to pose a hypothesis, but we really don't want to be telling our people who we're training exactly what to prove. You want to be open to what you may discover. And so we had a strong hypothesis that this thing would interact with the plasma membrane, but to Jaspreet's credit, this was not easy to prove, and he didn't just come back and say, oh yes, I made these things go to the plasma membrane. So we tried this for many months, actually. This is the Aster B tagged. You can see this nice reticular pattern. This is ER localization. This is a plasma membrane marker. And if you look here carefully, there really is no overlap. They're not really interactive. And at this point, you have to reevaluate your hypothesis. All right, so maybe they don't localize to the plasma membrane. Where do they localize? So then Jaspreet went back and redid this experiment with markers for every other compartment in the cell that we could think of, and he could never get any yellow. He could never get any localization. And so that caused us to reevaluate how we were doing the experiment. And if you think about it, you have to remember that in order for transport to move here, remember I explained these three different pools. Transport only happens when you have too much cholesterol in this pool. It turns out that we do almost all of our cell experiments in my lab in the absence of extra cholesterol in the media, because we study transcription factors that are activated by cholesterol, so we want to start with a baseline where you have no activity. So we take cholesterol out of the equation. So under the conditions we're doing these experiments, there wasn't any transport going on. And eventually we figured that out. And so the trick is you have to add cholesterol to the cells, and then when you do that, voila, the protein moves in response to the cholesterol. These are cholesterol sensors, in effect. And it's a switch, and it's a switch that knows when there's too much cholesterol in the plasma membrane, and when there is, the transporter goes to the plasma membrane to pick up that excess cholesterol. So you can see the colocalization here. And this happens very rapidly. So what the aster proteins do is facilitate the formation of something called an ER plasma membrane contact site. So in the cell biology world, one of the things that cell biologists are very interested in is membrane-membrane contacts between different organelles. It turns out all the organelles are in contact with each other, and this likely facilitates the movement of molecules and proteins and lipids back and forth. It had been known that there are ER plasma membrane contacts, and it had been postulated that they might be important in non-vesicular transport of cholesterol, but that this hadn't really been worked out. So this is a complicated image, but this really proves that the aster proteins are cholesterol-dependent ER plasma membrane contact site proteins. The green here is the aster, the blue is the ER, and the pink is the plasma membrane. So if you look here carefully, in the absence of excess cholesterol, the green aster protein is always in the ER tubules. It's not floating around. It doesn't ever leave the ER. It's always in the tubule. What happens when you add cholesterol to the cell is that these green labeled ER tubules move to the plasma membrane. They form, they abut, the two membranes abut. And now you have this three-part structure with the aster protein, the plasma membrane, and the ER, and this is where the cholesterol transport is likely to occur. And one sort of subtle cell biology point I'll make here is that not all ER plasma membrane contact sites are cholesterol dependent. Only the aster dependent contact sites are. So this is another ER plasma membrane contact protein called ECIT2. This protein is present at the plasma membrane regardless of whether there's cholesterol there, whereas the aster is clearly entirely cholesterol dependent. And you can have different types of contacts. This one has both the ECIT protein and the aster, but this one has only the aster. And what this result implies is that just making the contact is not sufficient to move the cholesterol. You need to bring the two membranes together, but then presumably in the middle of that membrane is the aster domain, which is bumping back and forth and actually moving the cholesterol. So you need the transporter at the contact site to move the cholesterol. So that's how we think the pathway works. The next problem that we wanted to try to solve, and to be completely honest about it, we haven't solved this mechanism 100% yet. We want to know what the switch is. These proteins go to the plasma membrane only when there's too much cholesterol there, excess cholesterol in the accessible pool. So how does the protein know when there's too much cholesterol there? Well, the protein binds cholesterol in its central domain, so you would immediately think that the cholesterol binding domain must be important in the cholesterol-dependent recruitment to the plasma membrane. And that's what we initially thought, and that turns out to be entirely wrong. It turns out that the domain that's responsible for the cholesterol-dependent switch is actually just this little N-terminal gram domain, which is the part that binds phospholipids. which was a complete surprise. So my daughter, who's 9, calls this the avocado picture. She loves this because avocado is her favorite food. And what this image shows is that just this little soluble gram domain by itself is cholesterol dependent. If you add cholesterol to the cell, this soluble cytoplasmic domain immediately goes to the plasma membrane. So that's able to sense the cholesterol. So how does that work? This just shows that this happens very rapidly. This is over the course of 15 to 30 minutes or so. You can see within minutes, the protein starts to move when you add cholesterol to the cell. So it's a very rapid switch. We've tried to model this in artificial systems. We're having some success doing that. Ultimately, we'd like to understand the structural basis and solve the crystal structure of that domain in complex with a membrane. The implication is that the gram-like domain sees some structure in the membrane which changes when the cholesterol concentration increases. And we think it's likely sensing a change in the presentation of the phospholipid, the phosphatidylserine itself. And here are some in vitro studies that sort of support that idea. If you look just over here on the right, what we're doing here is making a vesicle that we're asking, can that vesicle interact with a protein or not? And if it's in the pellet, which is the P here, that means the vesicle and the protein are interacting. And what this curve shows is that if you just put cholesterol in the plasma membrane and phosphatidylcholine together, the aster protein doesn't bind. But if you now put in phosphatidylserine, that allows the aster protein to sense increasing cholesterol concentration. So what we think happens is that the cholesterol concentration increase changes the presentation of the phosphatidylserine that the aster protein can now see. All right. So I've proven to you that the proteins bind cholesterol, they move them in a dish and in a test tube, but at the end of the day, I'm a physiologist, a mouse physiologist. I'm interested in how things work in an animal, in a living system. So we needed to test the idea that these were cholesterol transporters in an actual cell and then in an actual animal. First, we're going to test this in cells, and we're going to do this by adding excess cholesterol to a cell and then looking for a response to that excess cholesterol in the plasma membrane. The first thing that we did was to look at the SRBP pathway. So this is a transcription factor that lives in the ER, and it's activated by cleavage in the ER, which is controlled by cholesterol levels. So in order to regulate this pathway, the cholesterol needs to be getting to the ER. And what we could show is that in cells where aster expression is silenced, you have increased expression of the SRBP pathway in the ER, which implies that the cholesterol is not as effectively moving to the ER. So that's one measure. Another way you can do the experiment is to look for the ability of the cell to turn that free cholesterol into ester. You can give the free cholesterol to the plasma membrane and then ask, can the cell make cholesterol ester out of it? And in order to do that, it would have to get to the plasma, to the ER. And in the absence of the aster proteins, that doesn't happen. That's actually the LXR. I'll skip to this one. So this is the esterification experiment. In the absence of the aster protein, the rate at which you get cholesterol accumulation is significantly, cholesterol ester accumulation is significantly retarded. But those indirect measures imply that the cholesterol is not getting to the ER, but I really didn't prove that the cholesterol is actually backing up on the plasma membrane. How would we prove that? Well, your first thought might be, well, can't you just measure the amounts of cholesterol in the plasma membrane? Can't you just take the cell, fractionate it, isolate the plasma membrane, and look for changes in cholesterol content? But when you think about what I told you at the beginning, the plasma membrane is 30% cholesterol, but only a very small fraction of that is the pool that we care about. What you're going to see is if you fractionate the cholesterol in the plasma membrane and look for changes, is you're going to see nothing. That's because what you will be looking for is a change between 30% cholesterol and 31% cholesterol, which you'll never be able to measure. So you need a way to specifically assess this accessible cholesterol pool. And my colleague Arun Radhakrishnan at UT Southwestern has shown that there are these bacterial toxins, one of which is called anthrolysin, which binds cholesterol. And it turns out what they bind are specifically this accessible pool. My colleague Steve Young has adapted this protein for use in a technique called nanosims. I'm not sure if anybody has heard of that approach, but I will summarize it by saying nanosims you can think of as a cross between electron microscopy and mass spectrometry. What this instrument does is take a very small slide on which you have cells. You have a laser which goes across the slide and vaporizes the material and then sucks up what's coming off and puts it through a mass spec and asks what isotopes are there. And this is a way that you can get very high resolution imaging of where an isotopic label is in your sample. So what Steve did was to tag this ALO protein, which specifically binds accessible cholesterol, with an N15 probe. And if you do that, you get these beautiful images. So the purple here, what you're looking at in the purple, you're looking down on the top of a cell. We added this ALO protein to the cell. It doesn't get in the cell because it's a protein. It's on the surface of the cell, but it's binding the surface of the cell at an increased rate here because there's too much accessible cholesterol on the plasma membrane. So this experiment proves that in the absence of the aster protein, not only is the cholesterol not getting to the ER, but it's actually backing up on the plasma membrane. And this is one of the few ways that you can really see that. And finally, for the last few minutes, I'm going to talk about in vivo biology. I really care about what things do in a living organism. I want to remind you that there's something called lipoprotein metabolism, which moves lipids around the body. There are lipoproteins, including LDL and HDL. You probably all know LDL as the bad cholesterol, and LDL moves around and gets internalized into cells via the LDL receptor. HDL actually is different, and it's not really well understood how HDL cholesterol gets into cells. The HDL receptor is a protein called SRB1, which is highly expressed among other places in steroidogenic tissues, in tissues such as the adrenal gland and the ovaries and the testes that take cholesterol and turn it into steroid hormones. SRB1 does something interesting and distinct from the LDL receptor. It's not internalized. What SRB1 does is bind the HDL particle on the surface of the cell. Somehow it sucks out the cholesterol and puts it into the plasma membrane. And then it's been known that this cholesterol must move to the ER because other things are done with it by the cell, but it's never been shown how HDL cholesterol gets to the ER. It turns out that one of the three aster proteins, this one aster B here, is very highly expressed in steroidogenic tissues and very nicely has an expression pattern which overlaps with SRB1, the HDL receptor. And that suggested to us that the aster proteins might functionally be downstream of the HDL receptor in vivo. And we got confirmation of that when we generated mice which lack aster B. If you open the organisms up, there's one tissue that is the wrong color, and that is the adrenal gland. This is what a normal adrenal gland looks like. It's sort of pinkish like this because the tissue is full of lipid. Remember I told you that the adrenal cortex stores cholesterol ester so that it can use that cholesterol to make steroid hormones. You can clearly see in the absence of the aster B protein, the tissue is the wrong color, and that's because all of the lipid is gone. This is oil red O, which is a lipophilic stain. This is the cortex. All of this red here is lipid droplets, cholesterol ester droplets in the adrenal cortex. This is entirely missing of the in the absence of the aster protein. And this is a black and white effect. As someone who works on lipid metabolism, all the time we're mutating genes in mice and looking for very small changes in lipid levels in vivo. This is the most dramatic change we've ever seen in anything. All of the cholesterol ester in the cortex is completely gone in the absence of the aster B protein. So this tells us that this protein is absolutely essential for storing cholesterol ester in the adrenal cortex. In theory, if I hadn't told you anything up to this point, there could be two reasons why there's no cholesterol ester in the adrenal cortex. One is that, as I'm implying, the cholesterol is not getting to the ER, so lipid droplets can't be formed. But the other formal possibility is that maybe we somehow messed up the activity of the ACAT enzyme, and the the cholesterol can still get to the ER, but you just can't make lipid droplets out of it. And the way you distinguish between those proteins is to look at the activity of the ER SRBP pathway, which is responsible for making cholesterol. If the cholesterol was still getting to the ER, then the SRBP pathway should be off because the cell doesn't need to make any more cholesterol. But what we see in the Astor knockout mouse is that the SRBP pathway is massively elevated. So for reference, this is HMG-CoA reductase. This is the target of the statin drugs. Statins work in humans by controlling the level of this enzyme 1.1 or 1.2 fold. That's how you lower your cholesterol in humans, by very small regulation of this pathway. This protein is 10-fold upregulated in the absence of the Astor proteins. So that tells you that the ER cholesterol synthesis pathway is working overtime to make enough cholesterol to support steroid hormone synthesis, because it can't get the cholesterol from the HDL. That compensation likely explains why these mice aren't dead. The absence of cortisol is lethal. You need to have cortisol to survive. So the reason these mice aren't dead is they're making enough cholesterol to support their steroid hormone synthesis. But if you challenge them, you can clearly show that they're adrenal insufficient. One way to challenge a mouse and get it to make a lot of cortisol is to fast it, because cortisol raises blood glucose levels. You can clearly see that the ability to make corticosteroid in response to fasting is markedly impaired in the absence of the Astor protein. Now, if you remember, this phenotype then is the exact opposite of what we saw with the STAR-D1 knockout. Remember, the STAR-D1 knockout's job, STAR-D1's job is to put the cholesterol, move it from the lipid droplet into the mitochondria. And the knockout of that protein gives you a very striking phenotype, which tells you that's the function. The Astor knockout has the same striking phenotype, but it points to a different function. It points to an inability to get the cholesterol to the ER in the first place. So to summarize the pathway a little bit here, in the absence of excess cholesterol in the plasma membrane, the Astor proteins are anchored to the ER, but they're not making contact with the plasma membrane because there's no need to transfer cholesterol to the ER under that context. When HDL is needed to deliver cholesterol to the ER for steroid synthesis, or in other cells which have excess cholesterol accumulation in the plasma membrane, this triggers the recruitment of the Gram domain to the phosphatidylserine in the plasma membrane. The Astor domain then accepts cholesterol from the plasma membrane and moves it to the ER so that the ER can do its job. In the last couple minutes, I'll just mention where these studies are going from here on out. There are 3 different Astor proteins, A, B, and C. At this point, we think they all perform the same fundamental function of moving cholesterol from the plasma membrane to the ER. It's possible that they can move it from between other compartments as well, but at the very least, they all do it from the plasma membrane to the ER. We can show that there is, in fact, redundancy between the pathways in certain systems so far. Why do you need 3 of them? Well, it's sort of a rule in higher organisms that one way is that you expand your ability to regulate and get more complex systems is you take one gene and you duplicate it, and then what that allows you to do is to regulate those 3 genes differently in different places in different contexts. So the 3 Astor proteins are expressed highly in different tissues, and they're regulated by different transcription factors. And so that allows them to be plugged into different biological pathways in different contexts and move cholesterol in different settings. So for example, Astor A is abundant in the brain, B is abundant in steroidogenic tissues, and C is abundant in, among other places, the liver. Once you get to the point to have your own lab, you'll be writing NIH grants. And one of the things you have to say in an NIH grant these days, in order not to get a demerit on your review, is you have to say that you're going to consider sex as a biological variable. And that's an important thing because for many years, people in the field just studied male mice because it's simpler to look at one sex, and in general, male mice, at least in metabolism, tend to have some more severe phenotypes. So in general, people only looked at males, and that's inherently could, for obvious reasons, be a problem. Not to mention the fact, for least of which, it implies that somehow studying males is more relevant than studying females, which is not obviously true. But practically, it is challenging to do all of your mouse studies with 2 different sexes because it doubles the amount of work you have to do. Everybody has to say that in their NIH grant now. You have to say you're going to consider sex as a biological variable, and a lot of times, practically, you can't because you don't have the money or you can't generate enough mice to do both, and you do one or the other. But in practice, that turns out to be important, and this is an example of that. So if you compare male Aster-B wild-type het and knockout mice over the course of 10 weeks after weaning and look at their body weight, there's no difference. But if you look at the female mice, there's a big difference. And if you hadn't been paying attention, you would completely miss this. And in fact, there's even an intermediate phenotype in the heterozygote, which you almost never see. I study body weight in mice for a living, and almost always when you knock something out, the hets have no phenotype on effect on body weight. So there's clearly something interesting going on in females. And anybody want to guess what it is? So all of you who said estrogen are likely correct. So we think this is a defect in estrogen synthesis in the knockout, which parallels what we were seeing in the adrenal gland. The cortisol, the cholesterol is not being effectively imported into the estradiol synthesizing cells in the ovary. And you can see this by just taking a basal estradiol level, which is not a great way to do this in a bunch of mice. You can clearly see that the plasma estradiol is very low, even without doing the experiment properly and considering cycle and hormonal release. There's much less estradiol here. So this is a very strong defect in estrogen synthesis. So that's one example. I'm going to skip that. That's a little bit complicated. Another context that we're very interested in is when you think about it, so the adrenal gland is one organ which utilizes cholesterol, but there are lots of other organs in the body that play an important role in cholesterol metabolism. Two that you might think of off the top of your head would be the intestine, which is responsible for absorbing cholesterol from the diet, and the liver, which is responsible for packaging cholesterol and sending it to the rest of the body. It's important for making bile acids out of cholesterol. So those are two places where you really wouldn't think that a cholesterol transporter might have important roles. And so we've been spending a lot of time studying the intestine and the liver. The bad news is that both the intestine and the liver have more than one transporter. So knocking out just one asterin in either of those tissues actually doesn't give you a very big phenotype. You have to knock out more than one. And in the intestine, what you have to knock out is both B and C together. But when you do that, you can see if you measure the accumulation of cholesterol in the intestine, if you feed the mouse radioactive cholesterol, there's a defect in the rate at which that cholesterol is absorbed from the diet. And then if you look in the body, downstream of that, if you look in the liver, there's a marked deficit in the ability of that radioactive cholesterol to get all the way from the intestine into the liver in the knockout mouse. So clearly there's a systemic consequence of not having aster function in the intestine that we're decided to explore going forward, both in the intestine and the liver. So I will stop there. I want to emphasize that this was a project that really involved not only the work of talented people in my own group, including not the least Jaspreet Sandhu, who just left the lab to start his medical residency at Mass General, but he's destined to return to science when he's done with his clinical training. He was really instrumental in driving this project, and so there's no substitute for having driven and motivated people. That's why my laboratory has been somewhat successful, but I've been successful in recruiting talented people and to the extent that I can, not screwing them up, trying to set them on a good course. Other people in the lab also contributed, including Xu Xiao, Alessandra, JP, and Emily. The collaboration comes in with all of the various approaches that we took to studying this problem, including structural biology with my friend John in the UK, Steve Young, who's the expert in nanosims imaging here at UCLA, Alina Akonen, who's an expert cell biologist who helped us with a lot of the imaging to prove the cholesterol-dependent recruitment to the plasma membrane, and Dave Ford at St. Louis does all of our mass spec lipidomic analysis for us. So I will stop there, and if there are any questions and things that I glossed over or forgot to address, I'd be happy to answer. Thank you.
All right, thank you, Dr. Contanos, for a great presentation. We do have a few questions. We'll open it up for about 15 minutes or so, or until the questions run out. But we'll run through the ones that were posted a little bit earlier while you're giving your presentation. Here's first one from Chloe. Hi, Dr. Contanos. You mentioned earlier that the space within the aster proteins is not fully occupied by cholesterol, evidenced by the fact that some glycerol was present. You noted that this could have some important pharmaceutical implications. Could you elaborate on that?
Yes. One of the things we've been trying to do is to develop inhibitors of the astroproteins. You might also be able to develop activators, but at least at this point, you certainly could design a molecule that fits in there and blocks the ability of cholesterol to go in there. So we've been doing that. We've been having some success. And the pharmacological implication of that extra space is that you could occupy that with a small molecule. So we're trying to find compounds that are a little bit bigger than cholesterol on that end. So what sits in that at the start of that space is the 3-hydroxyl group on the cholesterol, the sterol ring. So down here in the corner is the 3-hydroxyl. If we've been putting extra groups on that to try to occupy that extra space to get more avid and selective inhibitors. So that's why it's relevant.
Okay. Just a disclaimer, we've got several schools within our biomedical science program. So you'll get cell signaling questions, you'll get neuroscience questions, you'll get pharmacology questions, all kinds of stuff. The next one is, in your high-fat diet, does it contain high cholesterol?
I'm assuming this was for that body weight experiments with the male versus female body weight effect. That experiment was actually on a high-fat diet, which just has high fat, not high cholesterol. But we routinely actually do normal chow, high fat and high cholesterol for everything, unfortunately. And the body weight phenotype we see on everything. For those of you who don't work in metabolism, it might not be obvious to you, but the reason why that makes sense that estrogen is protective is it's well known that female mice and female humans have some resistance to obesity that is estrogen dependent. So estrogen is protective against obesity. It regulates thermogenesis and body weight in multiple different ways. So it makes perfect sense that a deficit in estrogen production in a mouse would lead to a more male-like susceptibility to obesity.
Okay, thank you. That question was from you, and thank you. Another question from Inquao. Great talk. Are asters expressed in macrophages? If yes, do they regulate cholesterol homeostasis in macrophages? And do LXRs regulate asters? So maybe we break that up into sections. Are asters expressed in macrophages?
Yeah, so those are all great questions. So the answer is yes. The macrophages have a lot of B, but they also have a lot of A. The B form is the one that's regulated by LXR. And to be honest, this is another sort of teaching point about how biology works. When we first started this, we were actually looking for the transporter that moves from the lipid droplet to the plasma membrane. So one of the things that the LXR transcription factors will do is regulate cholesterol efflux from macrophages. And the way they do that is they upregulate a protein called ABCA1, which sits on the plasma membrane and pumps the cholesterol out of the cell. We were interested in how the cholesterol gets to the ABCA1 in the first place. So we were looking for the transporter that moves to the plasma membrane. And we ended up discovering the thing that moves in the other direction. That's sort of how biology works. You might consider or ask yourself, well, does that mean the transporter might actually be bidirectional? Could it be going both ways? We considered that, but at this point, we don't think so. And the reason for that is if you think about it, and the transporter is recruited to the plasma membrane only when there's too much cholesterol in the plasma membrane, how would it be able to make the contact to move in the other direction? Unless there's too much cholesterol in the plasma membrane, it's not going to be moving. It's not going to be recruited. So how it could move in a regulated fashion in the opposite direction is not clear. But we're still studying that, and we're definitely interested in how it regulates cholesterol homeostasis in macrophages. It might imply that it's more important for storage in a macrophage, and that's something we're definitely looking at.
Okay, I think the last part of that was, I don't know if you addressed it, do LXRs regulate asters?
Yeah, so they regulate the B form. So
they
regulate aster B, but not the other two.
Okay. Another question, how long was the fasting period you had the mice on to increase their corticosteroid levels?
I think that's just an overnight fast.
Okay.
Which, I mean, overnight, 12-hour, which is a 12-hour fast for a mouse is a very significant fast. They'll start to lose grams of body weight because mice have to eat all the time because they're very small and they expend a lot of energy. So that's a very substantial fast.
Okay. We have several more questions in the chat, and maybe some individuals might prefer to ask them in person, so you can always feel free to turn your camera on after we get through these next couple questions. Have you looked at Aster B activity in older female mice, and have you noticed differences in cholesterol transport in cells with PTEN mutations? Also, thank you for an interesting talk. That's from Angelo.
Not yet. We're still exploring the steroidogenesis phenotype. The ovary is a little bit more complicated to study than the adrenal gland because the estrogen synthesizing cells are much more dispersed and spread out. It's not like in the adrenal cortex, they're all in the same place and you can sort of very nicely look at it. The ovary is a lot more complicated. It's very small, and there's actually not that much molecular biology that's been done on the ovary. It's kind of a hard thing to study. There's no way to easily put it in a dish and model it. And there are some cell lines, but the cell lines are sort of imperfect. So we're still trying to really prove what the transport pathway is in the ovary. One approach that we're taking is to do transplants, to transplant a wild type ovary back into a knockout and vice versa, and see whether that you would predict if it really is estrogen production, an ovarectomy or a transplant would rescue the phenotype. So that's one approach we're taking.
Cool. Next question from Nofrisha. You mentioned that Aster can recognize phosphatidylserine only in the presence of excess cholesterol. Does that mean cholesterol changes how phosphatidylserine is presented in the plasma membrane? How does Aster recognize it?
Exactly. That's exactly right. That's exactly what I said. So that's what we think. You can imagine sort of, the tail or the head group is there in one position and there's cholesterol sitting next to it, sort of kinks it out in some way, and then the protein can recognize it. So that's what we'd like to solve. But in order to do that, we need the crystallized gram domain in complex with a membrane, or at least enough of the membrane that you can see what's interacting with the protein. So we're trying to figure out how that works, but that's the idea.
Cool. And I think this is the last one in the chat, also from you. Is there any BA metabolism change in the liver of BCD knockout mice?
There are changes in bile acid metabolism in some of the combinations of knockouts. So that's something we're definitely looking at. We don't think the intestinal absorption effect is due to bile acids, but there is an effect on bile acids, we think, to some extent. Which makes sense because they're made, in the cell, and so that would be an expected downstream consequence of not being able to appropriately move cholesterol from the outside to the inside. So that's something we're looking at for sure.
Awesome. Okay, so that's the last of the chat questions. What we'll do, since we're at the 131 mark, which is great timing on our part, exactly an hour. We'll end it here if you guys have to head out, but if you'd like to stick around maybe for the next few minutes, ask any extra questions, feel free to do so. But we'll officially sign off and we'll address a few more questions. Here's one that just came in. Among Aster A, B, and C, which one is more efficient in the steroid transport? What are the consequences of lacking Aster B in the mice model?
So that's sort of hard to measure. In a test tube, you don't obviously see a difference in rate of movement in a test tube. In a cell, but it's really hard to compare. What you would need is a particular cell that has exactly the same expression of all three to be able to compare and then set up an assay. And there's no naturally occurring cell that has exactly the same level of all three of them. What I can tell you is that it's clear from the mouse that at least in some contexts, they are functionally redundant. So I showed you the B-C double knockout in the intestine. The B and the C don't have the same phenotype. The B has a little phenotype, the C has no phenotype, and together they have a much bigger phenotype. So in the intestine, they're clearly redundant, at least in part. Which one is more effective? That's not clear, and that would be really hard to to establish unless you could prove the level of each of the protein in a particular cell, and that's really hard to do. But what you can address is their redundancy and whether you need to knock out, by asking whether you need to knock out more than one of them. And in general, with the exception of the adrenal gland and the ovary, most of the other tissues, you actually need to knock more than one of them out to get a striking phenotype. And that tends to be the rule in mouse work. You know, I showed you that if you don't do the right challenge, a lot of times you don't see a phenotype in a knockout. That LXR knockout mouse that I showed you at the beginning that gets overloaded with cholesterol, that's a striking phenotype. But if you just take an LXR alpha or even an LXR double knockout mouse and just feed it a chow diet for its life and leave it alone, you would never know there's anything wrong with it. Unless you feed it cholesterol, it doesn't have a phenotype. So part of the challenge in doing in vivo mouse work that you have to consider as you go through your training, it's really essential to design the right challenge to interrogate your model, because a lot of knockout mice don't have a phenotype at baseline, but if you poke them in the right way, then you uncover a phenotype. You know, when you think about it, a mouse in the vivarium, it was not the same as a mouse in the wild. It's not subjected to the same challenges and stresses that it needs to do. Or the Astra B knockout mice in the wild are probably lethal because in the wild they're being chased by cats and constantly stressed by, you know, being caught in people's cellars. And so, whereas in the vivarium, you know, they have their food brought to them, they have their water brought to them, people leave them alone, and so they're not really stressed. But in the wild, where they need to respond to stress and need to be much more active in their steroid synthesis, that steroidogenic deficiency might really, if not compromise their viability, compromise their reproduction, which explains why the protein is highly conserved. So just because you don't see a phenotype in your knockout mouse on a chow diet when you first look doesn't mean the protein doesn't do anything.
Great question. Thank you for that response. One more question. When you knock out aster in intestine, does it change how cholesterol is absorbed by MPC1, L1 in the intestine?
Yeah, so that's a great question. So we're very interested in figuring out what the relationship is between the two pathways. At the very least, they're both important. So obviously MPC1, L1 is important. But there's a synergy. So one way to measure the activity of the SRBP pathway, of the aster pathway, is to look at the regulation of the SRBP pathway genes. So if you knock out aster, the cholesterol doesn't get to the ER, and so therefore the SRBP pathway goes up. The SRBP pathway in the knockout mouse, if you take a ezetimibe, which inhibits MPC101, it's a cholesterol absorption drug which is developed for humans. The target is MPC101. The effect of that drug and the knockout of aster is synergistic on SRBP targets in the intestine. So they're both pathways are important, but whether they're parallel or they work in series, we don't know yet. My hypothesis is the asters are actually downstream of MPC101, and the MPC101 sort of binds the cholesterol and gets it localized in the plasma membrane, and then the asters move it after that. That's my working model at the moment, but we'll see.
Okay, thank you. That was another good question from Nafrisha. If there's any more questions, feel free to ask away, again, either in the chat or by unmuting and turning your video on. It's also encouraged. But Dr. Tontonos, I don't know if your schedule is pretty tight, but if you wanted to stick around maybe for just a couple more minutes, maybe till 1:40, to
Sure.
field any last second questions.
Yep. No, I'm fine.
Okay, but officially then, if you don't mind, thanking Dr. Tontonos either in the chat or with like little reaction functions, that would be also heavily encouraged. And I will officially thank you on behalf of Kent State and the School of Biomedical Sciences for a great talk and going into lots of cool details with the lipid pathway, and also the teaching moments in terms of collaborations and real world science. Those are also very informative and also very pertinent. So again, much thanks, much appreciation on behalf of Kent State and Biomedical Sciences.
Thank you for having me. Good luck to all of you. Stay safe.
Thank you. Okay, so people are flooding the chat with lots of thanks and praise. I did have one question. I'm in the human evolutionary biology program within the school, and I wonder if you've ever pontificated on what this might mean in terms of human evolution, if there's any implications along that, in terms of with astroproteins in particular and lipid transport and cholesterol transport.
Well, one thing you can certainly imply from this, I didn't mention this, but the proteins first arose in vertebrates, and they really arose along with lipoprotein metabolism. So SRB1 and the astroproteins, so the HDL receptor and the astroproteins appeared at the same time. And so when you think about it, if you're a unicellular organism like a yeast, you don't really have a lot of need to transport cholesterol from the outside of the cell and do other things with it, because you're not making steroids, you're not storing cholesterol from somewhere else. It's all sort of a compartmentalized system. And it seems the astroproteins really arose with systemic lipoprotein metabolism when you had cells in specific organs that are specialized to do specific things, bring cholesterol from the outside, synthesize something out of it, and then release it and package it, or package cholesterol into some other form. So you can sort of imply from the conservation that they're part of the machinery that evolved when multicellular organisms needed to move lipids around in a system and from one tissue to another. And all three of them are very highly conserved all the way down to fish. So all three of them have some reproductively essential function in vertebrates.
Cool. Have you or one of your bioinformatic-minded colleagues, have they looked at the sequence homology? I know you mentioned you don't like blasting things and looking at homology too often, but have you looked at are they well conserved?
It's not that I don't like it. I don't know how to do it, so
oh.
I let other people do that.
But it seems like they're very well conserved throughout vertebrate evolution. So that must have.
Yeah. So there aren't. Those are the only three. There aren't other members of the family. But what are it's clear from our work and others is this fold that we have a protein which has a lipid binding sort of pocket. You can form a fold like that in multiple ways with multiple different primary sequences. And there are proteins in lower organisms, including yeast, that have a fold that looks like the astrofold, but the protein's not homologous. So you can trace it back. You can see it's not a direct homolog, but that fold was used just like an EGF repeat or some other structural element gets mixed up and used in various proteins throughout evolution. So the fold itself is probably more ancient and is used in other proteins and other organisms. But the structure of the mammalian protein, where you have the transmembrane, this one fold, and then the gram, that doesn't exist in other species. But the element itself, is more common. And that's sort of what you, as an issue, you know that from being an evolutionary biologist. That's sort of how biology works. You have little structures of proteins that appear in multiple proteins over the course of evolution.
Yep, the great tinkerer, that's evolution. Okay, so I don't see any more questions in the chat. So again, thank you, Dr. Tontonos. We will officially end. I think Noel is the official moderator. But on behalf of the BMS Student Advisory Council, thank you for accepting our invitation, and again for a great presentation and fielding lots of great questions.
Okay, thank you all. Take care.
All right, thank you.